Picture, if you will, a small band of humans huddled around a fire somewhere in East Africa, perhaps 70,000 years ago. The flames flicker against the darkness. Beyond the circle of light, predators prowl. But the most dangerous enemies these people face aren't the ones they can see. In the blood of the woman nursing her infant, microscopic invaders are multiplying. In the lungs of the eldest hunter, bacteria are colonizing tissues he doesn't have words to name. In the water they'll drink tomorrow, parasites wait with patient, mindless hunger. These people cannot see their true adversaries. They have no concept of germs, no understanding of contagion, no microscopes to reveal the invisible warfare being waged inside their own bodies. And yet this unseen battle, repeated across millions of individuals over hundreds of thousands of years, would shape the very architecture of what it means to be human. Welcome to a journey through deep time, where death and survival wrote themselves into our DNA. Here's the paradox that lies at the heart of our story tonight. The diseases that slaughtered our ancestors by the millions are the very forces that built us. The plagues that wiped out entire populations left behind only those with the right mutations, the right immune responses, the right genetic accidents to survive. We are, quite literally, the descendants of the survivors, and we carry their hard-won adaptations in every cell of our bodies. Think about that for a moment. The reason you can fight off certain infections, the reason your immune system responds the way it does, the reason some of you watching might carry genetic variants that protect against malaria or tuberculosis, all of this was purchased at an almost incomprehensible price, paid in the suffering of people who died 10,000 years ago, 50,000 years ago, 100,000 years ago. How did our ancestors' agony become our advantage? How did mass death paradoxically strengthen our species? And what invisible hands were guiding human evolution through all those dark millennia before we even knew what evolution was? These are the questions we'll explore together tonight. For perspective, consider this. Modern medicine, antibiotics, vaccines, germ theory itself, has existed for roughly the last 150 years, a blink, a heartbeat in evolutionary time. But disease has been shaping humanity for millions of years. From the moment our distant primate ancestors climbed down from the trees, they entered a world seething with pathogens, bacteria that had been perfecting their deadly craft for billions of years, viruses that could hijack cells with terrifying precision, parasites that had evolved elaborate life cycles specifically to exploit mammalian hosts. For millions of years, humans had no defense except the ones written slowly into our genes, one generation at a time, through a process as brutal as it was effective. Those who couldn't survive simply didn't pass on their DNA. We are what remained. Now, before we venture deeper into this story, I'd like to ask something of you. Wherever you are in the world right now, whether you're in bed in California at midnight, having breakfast in Tokyo, winding down an afternoon in London, or anywhere else on this spinning planet, leave a comment below. Tell us where you're watching from, what time it is. There's something profound about knowing that people across the globe are all pondering the same deep questions about our shared human past, each of us connected through curiosity across time zones and continents. Tonight, we're going to trace the invisible thread that connects ancient plagues to your immune system. We'll meet the diseases that nearly wiped us out, and the genetic mutations that saved us. We'll explore how a devastating epidemic 40,000 years ago might be why some of you are resistant to HIV today. We'll discover why the Black Death of the Middle Ages was merely the latest chapter in a story that began when humans first gathered in groups large enough to sustain contagion. This is a story spanning the entirety of human existence, and it's a story best told slowly, carefully, with time to let the weight of deep history settle over us like a comfortable blanket. So settle in, get comfortable, dim the lights if you haven't already. This is going to be a long, contemplative exploration, the kind that's perfect for drifting off to sleep while voices from the deep past whisper their secrets into your dreams. Because tonight, we're not just learning about prehistoric diseases, we're learning about ourselves, about how suffering became survival, about how the invisible sculptors of evolution, bacteria, viruses, parasites, carved humanity into its current form, one generation at a time, with tools made of mortality and adaptation. Let's begin at the beginning, when disease first found humanity waiting, vulnerable and unaware, at the dawn of our species. For most of human existence, roughly 300,000 years of it, our ancestors moved. They followed herds, they tracked seasonal plants, they never stayed anywhere long enough to accumulate much of anything. Not possessions, not waste, and crucially, not disease. Picture a band of 30 hunter-gatherers crossing a landscape 15,000 years ago. They camp beside a river for a week, maybe two. Someone falls ill, perhaps from parasites in undercooked meat or contaminated water. The group moves on. The pathogen, if it needed a dense population to survive, dies out. The sick individual either recovers or perishes, but the infection ends there. The disease has nowhere to go. No sustained chain of transmission. The numbers simply don't work in the pathogen's favour. This was the human condition for the vast majority of our story. Occasional infections, yes. Injuries, certainly. But the catastrophic, sweeping epidemic diseases that would later define human history, they couldn't exist yet. We weren't important enough. From a pathogen's perspective, humans were a scattered, mobile, frustratingly sparse resource. We lived in groups too small to sustain transmission chains. We moved too frequently to create the filthy, crowded conditions that many diseases require. We were, in epidemiological terms, a terrible host species for the kinds of infections that would later shape civilizations. And then, somewhere around 10,000 years ago, everything changed. The Neolithic Revolution, that momentous transition from foraging to farming, is typically framed as humanity's greatest triumph. The birth of civilization, the foundation of everything we've built since. Agriculture allowed us to produce surplus food, which enabled population growth, which led to cities, writing, art, science, and every complex society that followed. But here's what the history books often gloss over. The Neolithic Revolution was also an epidemiological disaster. When humans stopped moving, diseases moved in. The transition happened differently in different places. In the Fertile Crescent around 9500 BCE, in China's Yellow River Valley around 7000 BCE, independently in Mesoamerica, the Andes, Sub-Saharan Africa, New Guinea. But wherever and whenever it occurred, the pattern was remarkably similar. Humans domesticated plants. They domesticated animals. They built permanent settlements, and with shocking consistency, their health deteriorated. Let's focus on what happened when we invited animals into our lives. Domestication seemed like such a practical innovation. Why chase wild aurochs across dangerous terrain when you could keep cattle in a pen? Why track migrating birds when you could raise chickens? Pigs converted waste into protein. Sheep provided wool and meat. Dogs helped with hunting and protection. Goats thrived on marginal land. Every domesticated animal solved a problem, and everyone became a biological time bomb. Because animals, it turns out, carry diseases, lots of them, and when you go from occasionally hunting an animal to living in constant, intimate proximity with herds of them, those diseases gain opportunities they never had before. This is how zoonotic transmission works. Pathogens jumping from animal hosts to human ones, and the archaeological and genetic evidence suggests it happened repeatedly, in waves, as agriculture spread. Cattle gave us measles. The genetic evidence is clear. Measles virus is closely related to rinderpest, a cattle disease. At some point, likely after humans had been keeping cattle in large numbers for generations, rinderpest mutated into a form that could infect humans. Early on, these infections probably burned out quickly, dead ends that never spread beyond the first victims. But as human settlements grew denser, as more people lived in close contact with cattle herds, the virus found its footing. Measles, one of the most contagious diseases in human history, is our evolutionary souvenir from domesticating cows. Smallpox, that ancient scourge that wouldn't be eradicated until 1980, likely emerged from a pox virus carried by rodents or livestock, possibly camels. The genetic relationships are complex, still being unravelled, but the timeline is clear. Smallpox as a major human disease couldn't have existed before agricultural settlements created the dense populations necessary for its transmission. Influenza jumps from birds and pigs, constantly shuffling its genetic material in those animal reservoirs, producing new variants that our immune systems have never encountered. The domestication of waterfowl and swine in ancient China and Southeast Asia gave flu a permanent staging ground for evolutionary experimentation. Tuberculosis, though its origins are more ancient and complex, found new purchase in the crowded, poorly ventilated dwellings of early farmers. The bacterium that causes TB can survive in cattle, and genetic evidence suggests multiple exchanges between bovine and human strains as domestication progressed. Even the common cold, that mundane annoyance, bears the signatures of animal origin. Several cold-causing viruses have close relatives in livestock species. Think about what this means. When a child today catches measles or flu, when a teenager comes down with strep throat, when an adult battles tuberculosis, these aren't just infections. They're evolutionary echoes of decisions our ancestors made 10,000 years ago. The moment we penned those first cattle, brought those first pigs into our settlements, nested those first chickens in our dwellings, we were signing an invisible contract with the pathogens they carried. We gained domesticated animals, they gained us. But the animals were only part of the problem. The settlements themselves became incubators. Imagine those first permanent villages. Excavation sites like Çatalhöyük in modern Turkey give us a window into what they were like. Houses built wall to wall, so tightly packed that people entered through holes in the roof. No streets, no sanitation systems. Waste accumulated. Food stores attracted rats. Water sources were contaminated by human and animal feces. Archaeological evidence from these sites tells a grim story. Bones show signs of infectious diseases. Dental remains reveal nutritional stress. Ironically, early farmers often had worse nutrition than hunter-gatherers, their diets now dependent on a narrow range of domesticated crops. Skeletons show growth disruptions, periods in childhood when disease or malnutrition temporarily halted development, leaving permanent marks in the bones. The population density created a new epidemiological reality. There's a threshold, different for each disease, but generally somewhere between several thousand and 500,000 people in regular contact, below which certain infections simply cannot persist. Measles, for example, needs a population of roughly 250,000 to 500,000 people in sustained contact to maintain continuous transmission. Below that number, the virus runs out of susceptible hosts. Everyone either dies, recovers and becomes immune, or never gets infected and the disease disappears. This is why measles couldn't exist in the Paleolithic. Hunter-gatherer bands of 30, even occasional gatherings of a few hundred, never came close to that threshold. Measles needed cities. It had to wait for us to build them. And we did. We built villages that grew into towns, towns that grew into cities. We created trade routes that connected distant populations, effectively enlarging the pool of susceptible hosts. We gathered for markets, festivals, religious ceremonies, mass events that facilitated transmission. We were, entirely unknowingly, constructing the perfect infrastructure for epidemic disease. Water, that essential resource that determined where humans could settle, became a disease vector. When you're nomadic, contaminated water is a local, temporary problem. You move on. The source is abandoned. But in a permanent settlement, where a spring or well serves hundreds or thousands of people, where that water is downstream from human waste, where the same source is used for drinking, washing and waste disposal, waterborne diseases flourish. The evidence appears in ancient remains. Coprolites, fossilized feces, recovered from early agricultural sites reveal an astonishing burden of parasitic infections. Whipworm, roundworm, tapeworm. These intestinal parasites thrive when human waste contaminates food and water supplies. Their eggs are remarkably durable, surviving in soil for years, waiting to complete their life cycle when accidentally ingested. Studies comparing coprolites from hunter-gatherer sites with those from early farming communities show a stark difference. The parasite load increases dramatically with settlement density. Some early agricultural populations show infection rates approaching 100% for certain parasites. Everyone was infected, all the time. It was simply the baseline condition of life. Malaria deserves special attention because its story is older, much older than agriculture. Plasmodium falciparum, the deadliest malaria parasite, has been with humans since our origins in Africa. Genetic dating suggests our ancestors were dealing with malaria long before we were even fully modern humans. Hundreds of thousands of years of shared history. And that timescale is crucial because malaria has had more time to shape human evolution than perhaps any other infectious disease. The parasite is transmitted by Anopheles mosquitoes, which breed in standing water. In forested environments, Malaria existed, but human populations were spread thin enough that transmission was sporadic. But when humans began clearing forests for agriculture, something happened. We created more open, sunlit areas with pools of standing water, perfect mosquito habitat. We increased population density, giving mosquitoes more, human targets in a smaller area. We domesticated animals that drew mosquitoes to human settlements. Agricultural societies in Africa, Southeast Asia, and eventually the Mediterranean and beyond created ideal conditions for malaria transmission, and malaria exerted tremendous selective pressure. The regions where malaria was endemic became evolutionary hotspots where any genetic variant that offered even partial resistance to the parasite would be favoured by natural selection. But we're getting ahead of ourselves. That's a story for the next section. For now, it's enough to understand that malaria represents the deep history of human-pathogen interaction, a relationship that predates agriculture but exploded with it. The archaeological evidence of this disease burden is written in bones and teeth. Paleopathology, the study of ancient diseases through physical remains, has advanced dramatically in recent decades, combining traditional skeletal analysis with molecular techniques. Tuberculosis leaves characteristic lesions on bones, particularly the spine and ribs. These lesions, called Pott's disease when they affect the spine, show the chronic, destructive nature of TB infection. Skeletons from Neolithic farming communities show these lesions with increasing frequency compared to earlier hunter-gatherer remains. The disease didn't just exist, it was becoming more common, more successful as a human pathogen. Leprosy, another chronic bacterial infection, leaves distinctive changes in facial bones and the bones of hands and feet. Evidence of leprosy appears in agricultural societies, particularly as trade networks expand and population density increases. Treponematosis, a family of bacterial diseases including syphilis and yaws, shows up in bone lesions with distinctive patterns. The exact origins and relationships between different treponematoses are still debated, but the evidence clearly shows these infections circulating in prehistoric populations. Even diseases that don't leave obvious bone changes can sometimes be detected through other evidence. Dental enamel hypoplasia, lines or grooves in tooth enamel, forms when growth is disrupted during childhood, often due to disease or malnutrition. Comparing teeth from hunter-gatherer and early farmer populations consistently shows more enamel hypoplasia in agricultural communities. Childhood in these early farming settlements was harsher, sickness more frequent, growth more often disrupted, and then there's the revolution in ancient DNA analysis. Over the past 2 decades, scientists have learned to extract and sequence DNA from ancient remains. Not just human DNA, but pathogen DNA as well. Fragments of bacterial and viral genomes can survive in bones and preserve tissues for thousands of years. With enough genetic material and sophisticated enough analysis, we can identify what pathogens infected ancient people, trace how those pathogens evolved, and even reconstruct their pandemic, spread. DNA from Yersinia pestis, the bacterium that causes plague, has been found in remains dating back to the Bronze Age, 3,000 years before the Black Death. Mycobacterium tuberculosis, the TB bacterium, has been sequenced from prehistoric remains, allowing researchers to trace how different strains evolved and spread. Even snippets of ancient viral DNA, though more difficult to recover, occasionally survive, offering glimpses into the viral diseases of prehistory. This molecular evidence confirms what the bones and coprolites suggest. Early agricultural societies were disease-ridden. The concentration of people, animals and waste created conditions where pathogens didn't just survive, they thrived and diversified. But here's where the story becomes more than just a tale of human suffering. Because diseases weren't just infecting humans, they were changing us. Every infection was a test, a sorting mechanism. Individuals whose immune systems happened to mount more effective responses survived and reproduced. Those whose immune defences were inadequate died, often before reaching reproductive age. Generation after generation, the population was being sculpted by selective pressure from pathogens. This is natural selection in its rawest form, not gradual adaptation to climate or food sources, but rapid, intense selection imposed by invisible enemies that reproduced thousands of times faster than their human hosts. While a human generation took roughly 20 years, bacteria could reproduce in 20 minutes, viruses even faster. Pathogens could evolve, mutate, and adapt on timescales that dwarfed human evolution, and yet humans evolved too. We had to. The alternative was extinction. The evolutionary arms race had begun. Pathogens evolved mechanisms to evade human immune responses. Humans evolved enhanced immune defences. Pathogens evolved countermeasures. Back and forth, an endless dance of adaptation and counter-adaptation played out across thousands of generations. Some of the results were straightforward improvements to immune function. Other outcomes were more complex, more costly. Defensive measures that provided protection against disease, but came with significant trade-offs. Genetic changes that saved lives in the short term, but created vulnerabilities in the long term. Evolution doesn't optimize for long-term health or comfort. It optimizes for reproductive success in the immediate environment. If a genetic variant helps you survive to reproduce in a disease-ridden agricultural settlement, natural selection favours it, regardless of what other effects it might have. Let's look at what we can actually measure in the archaeological record. Studies comparing skeletal remains from late hunter-gatherer populations with those from early agricultural communities in the same regions provide stark contrasts. In the Fertile Crescent, where agriculture first developed, skeletal evidence shows that early farmers were shorter than their hunter-gatherer predecessors. Height is a sensitive indicator of childhood health and nutrition, and the decrease suggests increased disease burden and nutritional stress. Their bones show more signs of infection, more indicators of anemia, more evidence of nutritional deficiencies. Similar patterns appear in early agricultural populations in China, Mesoamerica, and North America. The transition to agriculture is consistently associated with declining health indicators, at least initially, and at least for the majority of the population. Some individuals in these societies, likely elites with better access to resources, and perhaps less exposure to the dirtiest, most crowded conditions, may have fared better. But for the average person, farming meant more disease, harder work, and often shorter life. The life expectancy numbers are difficult to calculate with precision from skeletal remains, but the broad patterns are clear. Hunter-gatherers who survived infancy often lived into their 50s, 60s, sometimes longer. Early farmers faced higher childhood mortality and more episodic disease outbreaks that could decimate entire communities. So why didn't humans just abandon agriculture and return to hunting and gathering? Because by the time the health cost became clear, populations had grown beyond the carrying capacity of a hunting-gathering lifestyle. There were too many people to sustain through wild resources alone. Agricultural communities could support larger populations, even if those populations were, on average, less healthy. The surplus production from farming, despite its nutritional limitations and disease burden, could feed more people than hunting and gathering in the same area. It's one of history's bitter ironies. Agriculture allowed populations to grow, which made agriculture necessary, which caused disease to flourish, which began shaping human genetics in ways that would echo for millennia. The diseases of the Neolithic Revolution, these were the selective forces that began rewriting our DNA. Every epidemic was an evolutionary bottleneck. Every plague was a genetic filter. The humans who survived to become our ancestors did so, in part because they carried genetic variants that offered some protection, some advantage, some edge in the biological arms race against pathogens, and those genetic changes, accumulated over hundreds of generations of living with endemic disease, didn't just disappear when medicine advanced or sanitation improved. They're still with us, still in our DNA, still affecting our health, our immune responses, our susceptibilities to modern diseases. The agricultural revolution transformed the human relationship with disease from occasional encounters to constant coexistence. We went from visitors in the pathogen world to permanent residents. And that change, that fundamental shift in how humans lived, and what we were exposed to, in how natural selection shaped our populations, set in motion genetic changes that would ripple through our lineages for thousands of years. The microscopic enemies we met in those first agricultural settlements didn't just make our ancestors sick, they rewrote our evolutionary story. They changed what it meant to be human at a genetic level. In the next section, we'll explore exactly how that happened, how specific diseases drove specific genetic changes, how populations developed different immunological profiles based on their disease environments, how the evolutionary shields raised against ancient plagues still protect us, constrain us, and sometimes betray us today. Because those early encounters between humans and pathogens, that was just the beginning. The real story is written in our genes. Your genes remember things you don't. Hidden in the spiral staircase of your DNA are scars from battles fought thousands of years before your grandparents were born. Mutations that should have disappeared, persisting. Variations that seem harmful at first glance, thriving across entire continents. And all of it, every strange quirk, every unexpected pattern, tells the same story. Disease didn't just kill our ancestors, it transformed them. To understand how, we need to travel to West Africa. Picture the landscape, dense tropical forests, standing water, and in those waters, clouds of mosquitoes. Anopheles mosquitoes, each one a potential carrier of Plasmodium falciparum, the deadliest species of malaria parasite. For thousands of years, this microscopic killer has been one of the most powerful evolutionary forces in human history, and it left its signature written in blood. The sickle cell trait. You've probably heard of it. A genetic condition that causes red blood cells to warp into crescent shapes under certain conditions, blocking blood vessels, causing pain, organ damage, sometimes death. It's a textbook example of a genetic disease. Medical students learn about it in their first year. But here's what makes it remarkable, what transforms it from a simple genetic disorder into a window onto the entire mechanism of evolution. In malaria zones, this disease is actually a gift. If you inherit 2 copies of the sickle cell mutation, one from each parent, you have sickle cell disease, the full devastating version. But if you inherit just 1 copy, 1 mutated gene and 1 normal gene, you're a carrier. And carriers have something extraordinary, resistance to malaria. The parasite can't complete its life cycle in misshapen cells. The infection stalls. Children who would have died from cerebral malaria survive. Adults who would have been weakened by chronic infection stay strong. In regions where malaria kills millions, having 1 copy of this mutation increases your chances of survival by up to 90%. Think about that trade-off. In a population without malaria, the sickle cell mutation would disappear. Natural selection would eliminate it because individuals with 2 copies die young. But introduce malaria, introduce a disease so deadly that it kills before reproduction, and suddenly the mathematics change. Carriers thrive. They survive to have children. And those children, statistically, will include both carriers, protected from malaria, and a small percentage with the full disease. It's a cruel bargain, written in the currency of survival. This is what we call balancing selection. The mutation persists not despite its harmful effects, but because in the right environment, the wrong environment really, one filled with parasites, those harmful effects are outweighed by protection. Natural selection doesn't care about perfect. It cares about what works, and you can see it on a map. Plot the frequency of sickle cell trait across the world, and the pattern emerges like a revelation. Highest concentrations in sub-Saharan Africa, particularly West and Central Africa. Significant presence in the Mediterranean, Greece, Turkey, Italy, the Arabian Peninsula, parts of India. Every single hotspot aligns with one thing: historical malaria transmission zones. The overlap is so precise, so undeniable, that you can use genetic data to map where malaria was endemic thousands of years ago. But sickle cell isn't alone. Thalassemia, another blood disorder that reduces hemoglobin production, follows the same geographic pattern. Mediterranean coast, Southeast Asia, the Middle East. All malaria regions. G6PD deficiency, a genetic condition affecting red blood cell metabolism, shows the same distribution. 3 different mutations, 3 different mechanisms, 1 selective pressure. Malaria didn't just kill people, it edited entire populations. For perspective, these mutations arose independently, in different populations in different time periods. That's convergent evolution playing out in real time, in human populations driven by a single parasite. The signature of disease is so powerful, so consistent, that it shaped our genome in predictable, repeatable ways across continents. And malaria isn't the only disease that left its mark. Let's shift to medieval Europe, the Black Death, smallpox, wave after wave of epidemic disease crashing through dense populations, and hidden in that carnage, another mutation was spreading. This one so recent in evolutionary terms that we can trace its approximate origin date. The CCR5 delta 32 mutation. CCR5 is a gene that codes for a protein receptor on the surface of certain immune cells. Under normal circumstances, this receptor helps coordinate immune responses, but it has a vulnerability. It's also the doorway that HIV uses to enter cells. The virus binds to CCR5, uses it as an entry point, and begins its replication cycle. Except in people who have the delta 32 mutation, a 32 base pair deletion that renders the receptor non-functional. No functional receptor, no viral entry, no infection. Individuals with 2 copies of CCR5 delta 32 are essentially immune to most strains of HIV. Carriers, people with 1 copy, show significantly slower disease progression if infected. It's one of the most powerful genetic protections against a modern viral disease, but HIV only emerged in the 20th century. So why does this mutation exist at all? Because something else selected for it. The delta 32 mutation is almost entirely confined to European populations, with frequencies highest in Northern Europe, around 10 to 14% in some regions. That distribution suggests it arose relatively recently, perhaps 700 to 2,000 years ago, and then spread rapidly. Genetic modelling indicates that only a massive selective pressure, repeated deadly epidemics, could account for that speed of spread. The leading candidates: plague and smallpox. We know the CCR5 receptor plays a role in immune response to multiple diseases. Some research suggests that the delta 32 deletion may have provided protection against Yersinia pestis, the bacterium behind the Black Death, or against smallpox, which ravaged Europe for centuries. The hypothesis is still debated, but the pattern is undeniable. The mutation that offers HIV resistance today exists because our ancestors survived different epidemics entirely. Their survival is your protection. But here's where it gets even more fascinating. These individual mutations, sickle cell, thalassemia, CCR5 delta 32, they're just the visible peaks of a much deeper transformation. Because the genes most affected by disease pressure aren't the ones that code for blood cells or cell receptors. They're the ones that code for recognition, the HLA genes, human leukocyte antigens. If you want to find the most variable region of the human genome, the stretch of DNA where individuals differ most dramatically from one another, you look at the HLA complex. These genes code for proteins that sit on the surface of your cells like identification badges, presenting fragments of whatever proteins are being made inside. If a virus infects a cell, HLA proteins display viral fragments on the surface. Your immune system's T cells patrol constantly, checking these badges, and if they spot something foreign, a viral fragment, a bacterial protein, they trigger an immune response. The system only works if your HLA proteins can effectively present the specific pathogens you encounter. And here's the evolutionary pressure. Different HLA variants are better at presenting different pathogens. If everyone in a population had the same HLA type, a single pathogen that evades that type could wipe them out. Diversity is survival. And so, over hundreds of thousands of years, pathogen pressure has driven HLA genes to become the most polymorphic genes in the human genome. There are thousands of HLA variants circulating in human populations. Some are ancient, shared with chimpanzees, maintained across millions of years. Others are newer, region-specific, tailored to local disease environments. You can trace human migration patterns by tracking HLA diversity. Populations that faced unique pathogen pressures developed unique HLA profiles. It's evolution in fast-forward, selection happening so intensely that the signal is written into the most fundamental identification system your body possesses. And sometimes disease drives evolution in unexpected directions entirely. Lactase persistence, the ability to digest milk as an adult. At first glance, it seems unrelated to infectious disease. It's about nutrition, about agriculture, about the domestication of cattle and goats, and the cultural shift toward pastoralism. And all of that is true, but there's another layer. For most of human history, and indeed for most mammals, the ability to digest lactose, the sugar in milk, switches off after weaning. There's no evolutionary reason to maintain the enzyme lactase into adulthood when milk isn't part of the diet. But around 10,000 years ago, in several populations independently, mutations arose that kept the lactase gene active throughout life. These mutations spread with remarkable speed. In northern Europe, lactase persistence is now nearly universal. In parts of East Africa, among pastoralist groups, it's the norm. The traditional explanation emphasises the nutritional advantage. Populations with domesticated animals gained a rich source of calories and nutrition from milk, and those who could digest it thrived. But here's the disease angle. Clean water was scarce. Throughout human history, water sources have been vectors for disease. Cholera, typhoid, dysentery, all transmitted through contaminated water. Boiling water kills pathogens, but fuel was often limited. Fermentation helps, but it's not perfect. Milk, fresh from the animal, is sterile. It's a safe source of hydration in environments where water could kill you. Populations that could digest milk had an alternative to potentially contaminated water, and in dense settlements where waterborne diseases flourished, that alternative became life-saving. The selective advantage wasn't just nutritional, it was epidemiological. This is what makes evolutionary analysis so complex. Adaptations are rarely driven by single causes. Lactase persistence probably spread for multiple reasons. Nutrition, culture, disease pressure, all reinforcing each other, all creating an environment where the mutation thrived. And now, in an ironic twist, that ancient adaptation might be contributing to modern health challenges. Because here's the paradox. As we've conquered infectious disease, at least in wealthier nations, we've seen an explosion of autoimmune diseases, allergies, chronic inflammatory conditions. Asthma rates have tripled in some populations. Type 1 diabetes, multiple sclerosis, inflammatory bowel disease, all increasing, particularly in highly developed countries with excellent sanitation. The hygiene hypothesis proposes an explanation. Our immune systems evolved in a world full of pathogens. Constant exposure, constant challenges, constant recalibration. The absence of those challenges, the clean water, the antibiotics, the reduced parasite load, leaves immune systems untrained, overreactive, attacking harmless substances or even the body's own tissues. We've removed the evolutionary pressure that shaped us, and our bodies haven't adjusted. It's a reminder that evolution doesn't optimize for longevity or comfort. It optimizes for survival to reproduction in a specific environment. Change the environment too quickly, and you're left with mismatches. Bodies adapted for challenges that no longer exist, unprepared for the ones that do. But evolutionary mismatches aren't the only legacy of ancient diseases. Sometimes the protection we carry came from outside our lineage entirely. When our ancestors left Africa, around 60,000 to 70,000 years ago, they encountered populations that had been separated from the main human lineage for hundreds of thousands of years. Neanderthals in Europe and Western Asia, Denisovans in Eastern Asia. And instead of remaining separate, our ancestors interbred with them. We know this because fragments of Neanderthal and Denisovan DNA still exist in modern human genomes. If your ancestry is European or Asian, roughly 1 to 4% of your genome comes from these archaic humans, and some of those fragments are clustered around immune system genes. That's not random. Neanderthals and Denisovans had been living outside Africa for hundreds of thousands of years. They'd faced different pathogens, different disease pressures, and their immune systems had adapted accordingly. When modern humans arrived, carrying African-adapted immune genes, they suddenly encountered novel diseases, viruses and bacteria they'd never seen before. Interbreeding provided a shortcut. Instead of waiting for beneficial mutations to arise randomly, modern humans acquired pre-adapted immune variants from populations that had already solved the problem. It's horizontal gene transfer, but instead of bacteria swapping plasmids, it's human populations exchanging survival tools through sex. Studies have identified specific immune genes, particularly toll-like receptors, which detect pathogens, that modern humans inherited from Neanderthals and Denisovans. These genes are more common in non-African populations, precisely where you'd expect them if they were acquired through interbreeding and then maintained by natural selection, because they provided disease resistance. Our ancestors didn't just outcompete Neanderthals, they absorbed them, integrating their genetic innovations into our lineage. Some of what keeps you alive today was invented by a different species, and the genetic diversity extends even to blood. Your blood type, A, B, AB, or O, seems like a mundane medical detail. It matters for transfusions, for pregnancy compatibility, but beyond that it feels arbitrary. Except it's not. The ABO blood group system is ancient, maintained across millions of years, present in multiple primate species, and the reason all these variants persist is because they interact differently with disease. Blood type O individuals are more susceptible to cholera, but more resistant to severe malaria. Blood type A shows increased vulnerability to smallpox and certain strains of plague. Type B has its own profile of susceptibilities and resistances. No single blood type is optimal for all diseases, so populations maintain all the variants. It's another form of balancing selection, diversity preserved, because different environments favour different types. The geographic distribution of blood types reflects migration and disease history. Type O dominates in the Americas, possibly because ancestral populations face specific disease pressures during migration, or because founder effects reduce diversity. Type B is more common in Asia, Type A in Europe. These aren't neutral variations, they're echoes of epidemics we can't directly observe, written into the most basic classification of human blood. And all of this happened fast. When we talk about evolution, we usually think in terms of millions of years. Geological time, species branching slowly, accumulated changes requiring epochs. But disease doesn't work on geological time. Epidemics work on human time, years, decades, a handful of generations. And when an epidemic is deadly enough, it can shift the genetic composition of a population in breathtakingly short periods. Consider the Black Death again. It killed between 30 and 60% of Europe's population in just a few years in the mid-14th century. That's not gradual attrition. That's intense selection. Any genetic variant that provided even marginal protection would increase in frequency dramatically, simply because individuals without it died at higher rates. Recent studies analysing DNA from plague victims buried in mass graves show that survivors were more likely to carry specific immune gene variants that became more common in European populations after the plague. The epidemic didn't just kill, it filtered. It removed lineages, preserved others, and in doing so altered the genetic trajectory of entire nations. This is selection happening at the speed of disaster. The same pattern appears with smallpox in the Americas after European contact. Indigenous populations with no prior exposure died at catastrophic rates, up to 90% in some communities. The survivors, the ones whose immune systems mounted effective responses, became the ancestors of subsequent generations. The genetic composition of Native American populations today reflects that winnowing, that brutal sifting. It's a sobering thought. Some of the genetic variants you carry exist because your ancestors survived plagues that killed their neighbours. Your genome is a survivor's inheritance. And these changes weren't just individual or even familial. They were population level, transformations that influenced migration, settlement patterns and cultural development. Populations that developed genetic resistance to local diseases could expand into regions that remained deadly to outsiders. The geographic distribution of malaria resistance correlates with historical patterns of settlement and agriculture in tropical regions. The spread of lactase persistence tracks with pastoralist expansion across grasslands. Immune gene diversity maps onto trade routes, conflict zones, places where populations mixed and diseases spread. Disease didn't just shape our genes, it shaped where we could live, how we moved, who we became. And here's the unsettling truth. This process hasn't stopped. It's still happening. HIV, tuberculosis, malaria, they're still selecting, still pushing human evolution forward. The difference is that we've developed tools to resist. Medicine, public health, technology. We've slowed the process, insulated ourselves from the harshest selection pressures. But the fundamental dynamic remains. Pathogens evolve. We evolve in response. It's a dance that began when the first replicators emerged billions of years ago, and it continues in your body right now, as your immune system patrols for threats, guided by genes shaped by battles older than memory. These genetic shields, sickle cell, CCR5, Delta 32, HLA diversity, all of them, are testament to a truth we sometimes forget. We are not separate from nature. We are not immune to evolutionary forces. We are products of them, shaped by them, defined by them, and the diseases that carved these protections into our DNA didn't discriminate. They struck populations, not individuals. They created pressures so intense that entire lineages were transformed. But those transformations had consequences beyond the genetic. They influenced who survived, where populations could expand, which cultures thrived. Because when disease rewrote our DNA, it didn't just change biology, it changed history. Picture, for a moment, a village, not today, not even 1,000 years ago. Go back further, 7,000 years. A settlement of perhaps 50 people, clustered together in mud brick dwellings along a fertile riverbank. Children play near cooking fires, adults tend crops and animals. Life is short, brutal in ways we can barely comprehend, but it's life. Then one person begins to cough. Within weeks, a third of the village is dead. Another third is marked forever, scarred, weakened, changed. The survivors bury their dead in shallow graves and try to understand what invisible force just tore through their community, like a predator they couldn't see, couldn't fight, couldn't outrun. This wasn't a rare tragedy. This was prehistory. And this scene, or something very much like it, played out across continents, across millennia, thousands upon thousands of times. The great plagues of our ancient past weren't aberrations. They were the baseline of human existence. Let's talk about humanity's oldest companion. Not fire, not tools, not language, tuberculosis. In 2008, researchers discovered something remarkable in the Eastern Mediterranean. Human remains, dated to approximately 9,000 years ago, showing unmistakable skeletal lesions characteristic of tuberculosis infection. The bones told a story written in calcium and decay, evidence of a disease that had already been with humans for millennia by that point. 9,000 years. Think about that time frame. The pyramids of Egypt weren't built until about 4,500 years ago. Rome wouldn't rise for another 6,000 years after this person died. And yet here was tuberculosis, already ancient, already embedded in the human experience. We call it the White Plague, the captain of all these men of death. It has more names across more languages than almost any other disease because it has touched every culture, every population, every corner of humanity's journey. And unlike some diseases that come in waves and then vanish, tuberculosis never left. It's the longest pandemic in human history, one that's still ongoing. The bacterium responsible, Mycobacterium tuberculosis, is a remarkably successful organism. It spreads through the air we breathe, targets the lungs we need to survive, and can remain dormant in a human body for decades before striking. In prehistoric populations with no treatment, no understanding of contagion, no way to isolate the sick, tuberculosis would have been catastrophic. Archaeological evidence suggests that in some ancient populations, up to 20% of adults showed signs of TB infection in their bones. 20%. That's not counting those who died too quickly for the disease to leave skeletal markers, or those who succumbed to the infection without bone involvement. The actual infection rate was almost certainly far higher. Tuberculosis killed slowly. That's part of what made it so devastating. An infected person could spread the disease for months, even years before dying. They'd cough, they'd weaken, they'd waste away, hence another name, consumption. And all the while, everyone around them breathed the same air, shared the same spaces, became exposed to the same invisible killer. But here's what's truly staggering about tuberculosis. We can trace its evolutionary relationship with humans through genetic analysis. The bacteria co-evolved with us. Different strains emerged as human populations spread across the globe, diversifying alongside our own genetic diversity. TB didn't just happen to us. In a very real sense, we grew up together. Now, let's shift to a disease that makes tuberculosis look slow: smallpox. If tuberculosis was humanity's longest companion, smallpox was perhaps its most terrifying. The pockmarked faces that appear in artwork from ancient Egypt, India and China aren't artistic license. They're documentary evidence of a disease that killed approximately 30% of those it infected and left survivors permanently, scarred, marked for life by an encounter with a virus that rewrote the human story. The story of smallpox begins, oddly enough, with rodents. Genetic analysis has traced the variola virus, the agent of smallpox, back to a common ancestor shared with other pox viruses found in animals. Somewhere, probably in Africa, probably between 10,000 and 20,000 years ago, a pox virus made the jump from animals to humans. Perhaps through hunting, perhaps through the early domestication of animals, perhaps simply by being in the wrong place at the wrong time. Once it established itself in human populations, smallpox became something unprecedented. The virus spread through respiratory droplets and direct contact. It could survive on surfaces, on clothing, even in dried scabs for weeks. An infected person would develop fever, followed by a distinctive rash that progressed to raised bumps, then fluid-filled pustules that eventually scabbed over. The entire process took weeks. The suffering was immense. But the real devastation of smallpox came from a particular characteristic. It granted immunity. If you survived smallpox, you'd never get it again. This created a terrible dynamic in human populations. In areas where smallpox was endemic, always present, it primarily killed children, constantly burning through each new generation. Adults walked around marked by scars but protected by immunity. The disease needed fresh victims to survive, and it found them in every newborn child. In populations that had never encountered smallpox, the devastation was apocalyptic. When the disease finally arrived in the Americas in the 16th century, carried by European colonizers, it killed an estimated 90% of some indigenous populations. That wasn't a prehistoric event, but it gives us a window into what must have happened repeatedly in our ancient past, when smallpox first reached a new population, when it jumped from one isolated group to another, when trade routes opened and connected communities that had never shared diseases before. The prehistoric impact of smallpox is harder to trace than tuberculosis because it rarely left skeletal evidence. But we know it was there. We know it shaped populations. We know it selected for specific immune responses that we still carry today. The scars weren't just on faces, they were written into our DNA. Then there's a disease that challenged not just bodies, but the very fabric of social organisation: leprosy. Mycobacterium leprae, the bacterium that causes leprosy, is one of the slowest acting infectious diseases known to humanity. It can take 5 to 20 years for symptoms to appear after infection. When they do, the disease attacks the skin and peripheral nerves, causing distinctive lesions, loss of sensation, and progressive damage that can lead to severe disfigurement. But here's what made leprosy unique in ancient times. It was visible, it was progressive, and it was terrifying. Archaeological evidence places leprosy in human populations for at least 4,000 years, possibly much longer. A 4,000-year-old skeleton from India shows clear signs of the disease. References to what might be leprosy appear in ancient texts from Egypt, China, and India. The disease was old even when recorded history began. The response to leprosy fundamentally shaped human social structures. This was the disease that taught humanity the concept of quarantine, a word that wouldn't be coined for thousands of years, but a practice that began in prehistory. People with leprosy were separated from their communities, forced to live apart, marked as unclean, untouchable, fundamentally other. This wasn't just cruelty, though it certainly was cruel. It was the first fumbling attempt at public health policy. Communities observed that something about proximity to diseased individuals seemed to spread disease. They didn't understand transmission. They couldn't see bacteria. They invented religious and moral frameworks to explain suffering, but they also isolated the sick, and in doing so they created one of humanity's most enduring social patterns. The stigma of leprosy persisted for thousands of years, long after the actual danger was understood to be minimal. Leprosy is actually one of the least contagious infectious diseases. It requires close, prolonged contact to spread. But the fear it generated shaped laws, religions, and social hierarchies across the ancient world. And it was far from the only plague writing its story into human bones. In 2015, geneticists made a discovery that rewrote our understanding of epidemic disease. They extracted DNA from human remains in Bronze Age burial sites across Eurasia, remains dating back 5,000 years. The DNA they found belonged to Yersinia pestis, plague, the Black Death, the disease that killed perhaps half of Europe's population in the 14th century. The pandemic that everyone associates with medieval horror, with mass graves and desperate prayers and the collapse of civilizations. It was already old 5,000 years ago. The Bronze Age specimens contained a form of Yersinia pestis. that predated the better-known medieval strain by thousands of years. These ancient bacteria were slightly different. They lacked some of the genes that made the medieval plague so explosively deadly, but they were plague nonetheless, and they were killing people in waves that left mass burial sites across the ancient world. Here's what fascinates researchers. These prehistoric plague epidemics might explain some of the major population declines visible in the archaeological record. Around 5,000 years ago, we see significant demographic changes across Europe and Asia. Settlements that had been growing suddenly shrank. Some areas became depopulated, cultural patterns shifted dramatically. Were these the fingerprints of plague? We can't know for certain, but we now know that plague was present, was spreading, was evolving. And we know that later versions of this same bacterium would become the single most deadly infectious disease in European history. The capacity for catastrophe was already there, lurking in ancient burial grounds, waiting to be discovered by researchers with modern genetic tools. Plague represents something crucial in understanding prehistoric disease. We're just beginning to uncover the full story. Every new genetic analysis, every excavated burial site, every ancient DNA sample rewrites what we thought we knew. The diseases that shaped our ancestors weren't just the ones that left obvious skeletal evidence. Some of the most devastating plagues might have killed so quickly, so completely, that they left almost no physical trace. Consider typhoid and typhus, two different diseases with confusingly similar names, both caused by bacteria, both spread in conditions where humans live in close proximity with poor sanitation. These were the camp fevers, the diseases of armies and settlements and anywhere people gathered in numbers. Typhoid spreads through contaminated water and food. Typhus spreads through lice, those tiny parasites that thrived in the unwashed clothing and bedding of prehistoric humans living in close quarters. Neither disease leaves reliable skeletal markers, yet we know from historical records that once writing began, these diseases were everywhere. They killed soldiers before battles could be fought. They decimated cities during sieges. They ravaged trade caravans and pilgrimages. They were so common that they were considered almost inevitable consequences of large-scale human gathering. Were they present in prehistory? Almost certainly. As soon as humans began living in permanent settlements of more than a few dozen people, we created the perfect conditions for these diseases to thrive. Poor sanitation, close quarters, limited understanding of hygiene, no treatment options. The ingredients were all there, and bacteria are opportunistic by nature. But here's the unsettling truth. Typhoid and typhus might just be the beginning. How many diseases have we lost to time? Think about what we've discovered so far. Tuberculosis, identifiable by bone lesions. Smallpox, traceable through genetic analysis and historical descriptions. Leprosy, visible in skeletal remains. Plague, recoverable through ancient DNA. These are the diseases we can find, can prove, can document with evidence that survived thousands of years. What about the diseases that killed everyone too quickly for bone changes to occur? What about the viruses that don't leave genetic traces in teeth and bones? What about the epidemics that struck small, isolated populations and vanished with them, taking their pathogens into extinction before they could spread further? Population genetics offers tantalizing hints. We can see population bottlenecks in human DNA, moments when the genetic diversity suddenly narrowed, suggesting that large numbers of people died in a relatively short period. Some of these bottlenecks align with known climate events or migrations. Others remain mysterious. Could they be plague events? Lost pandemics that killed so many people so quickly that they nearly erased entire genetic lineages? We may never know. But the absence of evidence isn't evidence of absence. The diseases we've discovered are likely just a fraction of the infectious catastrophes that shaped prehistoric populations. For every plague we can name, there might be 10 that remain forever anonymous. And disease didn't just kill, it moved populations. Human migration patterns show curious gaps and detours. Archaeological evidence reveals populations that suddenly abandoned regions they'd inhabited for generations. Genetic analysis shows groups that seem to avoid certain geographic areas for thousands of years, only to reoccupy them later. Were they fleeing disease? Imagine you're a member of a prehistoric community. A neighbouring village just a few days' journey away has been devastated by an unknown sickness. Half the people died within weeks. Survivors speak of fever, of weakness, of something invisible that killed indiscriminately? Would you stay in the area, or would you move, gathering your possessions, your family, and walking away from the invisible threat? This wasn't paranoia. This was survival strategy. Communities that fled endemic disease zones might have been the ones who survived to become our ancestors. Those who stayed, who couldn't move, who tried to weather the storm, might have been the genetic dead ends, the lineages that didn't make it to the present day. Disease shaped the map of human settlement as surely as mountains and rivers did. Certain regions became known as death zones, places where invisible forces killed outsiders who arrived. We now recognise many of these as areas where endemic diseases had reached equilibrium with local populations. Locals had genetic resistance or childhood exposure that granted immunity. Newcomers had neither, and they died in numbers that would have seemed miraculous to those who survived. Trade routes adjusted around disease zones. Migrations detoured away from known danger areas. Some of the most fertile, resource-rich regions remained sparsely populated, not because humans didn't try to live there, but because those who tried didn't survive long enough to establish permanent settlements. And as if the diseases themselves weren't enough, climate change kept introducing new threats. The Earth's climate has never been stable. Over the past 20,000 years, we've seen dramatic warming as ice sheets retreated, periods of cooling and drought, shifts in rainfall patterns and vegetation zones. Each environmental change redrew the map of where animals lived, where insects thrived, where water pooled and stagnated. And with each change came new diseases. As glaciers retreated and forests expanded, humans moved into regions that had been uninhabitable for thousands of years, but they weren't the first to arrive. Rodents, birds, insects, all carrying their own complement of bacteria, viruses and parasites, were already there. Each new environment brought humans into contact with new pathogen reservoirs. A warming climate allowed disease-carrying insects to expand their ranges northward. Mosquitoes carrying malaria, ticks carrying various bacterial infections, flies carrying trypanosomes. As humans followed resources into these newly accessible regions, they walked directly into disease environments they'd never encountered. Conversely, climate deterioration could concentrate humans and animals into shrinking habitable zones, creating the perfect conditions for cross-species disease transmission. Drought would force both humans and wild animals to congregate around the same dwindling water sources. Cooling temperatures would push populations south into regions already occupied, creating conflicts and creating opportunities for pathogens to jump between populations that had been separated for millennia. Every environmental shift was an epidemiological dice roll. Sometimes populations got lucky, sometimes they didn't, and the ones who didn't often vanished from the archaeological record, leaving only subtle traces in our collective DNA. But perhaps the most haunting aspect of prehistoric disease is this: childhood. Modern archaeology has revealed something that's difficult to confront emotionally, but impossible to ignore factually. In most prehistoric populations, more than half of all children died before reaching their 5th birthday. More than half. Look at the burial sites. Count the small graves, the tiny skeletons, the abbreviated lives. The numbers are overwhelming. Infant mortality wasn't just high, it was the dominant reality of early human existence. Having a child wasn't a guarantee of passing on your genes. It was a gamble, and the odds were stacked against survival. The majority of these deaths were from infectious disease, respiratory infections, gastrointestinal diseases, fever of unknown origin. Prehistoric children faced constant microbial assault, and their immature immune systems often couldn't mount an adequate defence. This created a brutal selection pressure. Children who survived to adulthood weren't just lucky. They were genetically equipped to resist the disease environment of their particular region and time. They'd weathered the storm that killed their siblings and playmates, and they'd carry forward the genetic variants that had saved them. Every prehistoric community was, in essence, a laboratory of natural selection where disease was the primary selective force. The children who lived to become our ancestors weren't necessarily the strongest or the smartest or the fastest. They were the ones whose immune systems happened to have the right combination of genetic factors to fight off the specific diseases circulating in their environment. Think about the cruelty of that system. Think about the sheer number of genetic lineages that simply ended, not because of predators or accidents or violence, but because of invisible microorganisms that killed children before they could have children of their own. And think about the cumulative effect. Individual epidemics were devastating. We can imagine the horror of a smallpox outbreak, the slow agony of tuberculosis, the sudden collapse of entire communities when plague arrived. Each disease was a catastrophe in its own right, a tragedy that played out in the bodies of real people who suffered and died without understanding why. But zoom out, look at the bigger picture. The cumulative effect of all these diseases, operating continuously across thousands of years, across countless human populations, created evolutionary pressure unlike any other force in our development. Predators could be avoided, climate could be adapted to, food scarcity could be mitigated by migration. But disease was everywhere, constant, unavoidable. It didn't just kill individuals, it shaped the very structure of our immune systems, selected for specific genetic variants, drove evolutionary adaptations that we still carry today. Disease created the biological arms race that would define what it means to be human, and that arms race, that desperate ongoing struggle between human biology and microbial evolution, that's where our story goes next. Your body is, at this very moment, engaged in a war. Not a metaphorical war, a real one. Microscopic invaders probe your defences every second of every day. Bacteria seeking entry through a microscopic cut, viruses drifting on the air you breathe, fungi colonising the moisture between your toes. And against this endless siege, your immune system stands guard. Not as a single fortress wall, but as a layered, adaptive, learning defence network that has been refined across hundreds of millions of years. This is the story of evolution's most sophisticated arms race, one where the stakes were nothing less than survival itself. Picture, if you will, the earliest multicellular organisms, simple creatures drifting through ancient seas, vulnerable to every bacterium and primitive virus that found their way through the cell membrane. These organisms needed protection, and evolution provided it, but not all at once, not as a gift. The immune system we possess today wasn't designed. It was built layer by layer through countless generations of trial, error, death, and unlikely survival. The foundation came first. What we now call the innate immune system, a collection of immediate broad-spectrum defences that respond to threats within minutes or hours. This ancient system evolved over 600 million years ago, long before our vertebrate ancestors even existed. It operates on a simple principle: recognise patterns common to many pathogens and attack them without hesitation. Think of it as the body's first responders. Neutrophils that engulf bacteria whole. Natural killer cells that destroy virus-infected cells before the infection spreads. Complement proteins that punch holes in microbial membranes. Macrophages, literally big eaters, that patrol tissues like sentries, consuming anything that doesn't belong. This system is fast, brutally effective against most threats, and completely, utterly unable to learn. That limitation might seem like a fatal flaw, but for hundreds of millions of years, it was enough. The innate immune system protected our distant ancestors through the Cambrian explosion, through the colonization of land, through the rise of early vertebrates. But as life grew more complex, so did the pathogens that preyed upon it. Viruses evolved mechanisms to hide from pattern recognition. Bacteria developed molecular camouflage. The arms race intensified. And then, roughly 500 million years ago, something remarkable happened in the ancestor of all jawed vertebrates. A genetic accident, possibly a viral insertion ironically enough, created the foundation for adaptive immunity. The immune system that could learn, that could remember, that could become more effective each time it encountered a specific threat. This was revolutionary, game-changing in the truest sense, because now the immune system wasn't just responding to pathogens, it was studying them. The adaptive immune system operates through B cells and T cells, specialized lymphocytes that can recognize virtually any molecular pattern through an almost miraculous mechanism of genetic recombination. Each B cell produces antibodies with a unique shape. Each T cell carries receptors that bind to a specific molecular signature. And here's the extraordinary part. Your body can generate hundreds of millions of different antibody shapes through random genetic shuffling, creating a recognition library vast enough to identify threats that have never existed before. When a B cell encounters its specific antigen, the molecular signature it was randomly designed to recognize, it begins to multiply rapidly. Within days, that single cell spawns an army of clones, all producing the same antibody, flooding the bloodstream with weapons precisely calibrated to that exact invader. But the real genius lies in what happens next. After the infection clears, most of these cells die off, but some remain. Memory cells that persist for years, sometimes decades, sometimes a lifetime. Waiting, ready to respond instantly if that pathogen ever returns. This is why you typically only get chickenpox once, why vaccines work, why your immune system becomes a personalised archive of every pathogen you've ever successfully fought off. The evolutionary advantages were immediate and profound. Populations with adaptive immunity could survive in pathogen-rich environments that would have decimated their ancestors. They could live in permanent settlements where disease spread more easily. They could travel, encountering novel infections, and return with resistance rather than death. But this power came with costs, profound ones that we still pay today. Let's talk about fever. That miserable, aching, shivering experience that makes you feel like death might actually be preferable to consciousness. Fever is ancient, not just in evolutionary terms, though it is at least 600 million years old, but in the sense that it represents one of the oldest deliberate responses to infection. Even cold-blooded reptiles seek out warm rocks when infected, behaviourally inducing a fever. Fish in aquaria will swim to warmer water when sick. The response is fundamental, primal, nearly universal among vertebrates, and it comes at an extraordinary metabolic cost. For every degree Celsius your temperature rises, your metabolic rate increases by roughly 10 to 13%. A fever of 39 Celsius, about 102 degrees, means your body is burning energy at a rate 13 to 25% higher than normal. For a sick person, already weakened, already struggling, this is an enormous investment. So why do it? Why did evolution preserve such an expensive response? Because it works. Fever inhibits pathogen reproduction. Most bacteria and viruses have temperature ranges where they replicate optimally, usually right around normal body temperature of 37 degrees. Raise the heat by even 2 or 3 degrees, and their replication slows dramatically. Some enzymes they depend on become unstable. Their metabolic processes falter. Meanwhile, fever enhances immune function. Your neutrophils become more mobile at higher temperatures. Your T cells proliferate more quickly. The production of antiviral interferons increases. Heat shock proteins, which help cells survive stress, become more active. It's a calculated gamble written into our genes. Burn energy now, suffer in the short term, but gain a crucial advantage against invaders that would otherwise overwhelm us. For a pregnant woman with a dangerous infection, or a child fighting off pneumonia, or a hunter-gatherer with sepsis from a wound, that extra degree or two of temperature might represent the difference between recovery and death. But here's where evolution shows its ruthless pragmatism. Fever can also kill you. Above 40 Beyond, proteins begin to denature. Brain damage becomes a real risk. The treatment becomes as dangerous as the disease. This is the first glimpse of a pattern we'll see again and again. The immune system didn't evolve to make you comfortable. It evolved to keep you alive long enough to reproduce. If that means days of agony, weeks of fever, permanent scarring from inflammation, these are acceptable trade-offs in evolution's cold calculus. Which brings us to inflammation. That redness, swelling, heat, and pain that accompanies even minor injuries and infections. Inflammation is protective. Fundamentally, vitally protective. When tissues are damaged or infected, blood vessels dilate, becoming more permeable. Fluid leaks into surrounding tissues, carrying immune cells to the site of injury. Neutrophils arrive first, within minutes, engulfing bacteria and debris. Macrophages follow, coordinating the response and beginning the cleanup. Chemical signals called cytokines orchestrate the entire response, calling in reinforcements, activating different immune cells, ramping up the intensity of the attack. This acute inflammation, sharp, intense, time-limited, has saved countless lives across evolutionary time. It prevents minor infections from becoming systemic ones. It walls off damage, preventing spread. It initiates healing. But inflammation is a double-edged sword, and the edge that cuts inward can be just as deadly as any pathogen. In the ancient world, most infections either killed quickly or were cleared quickly. The immune response could afford to be dramatic, overwhelming, even destructive to surrounding tissue, because the threat was immediate and the response would be brief. But our modern world presents a problem evolution never anticipated: chronic activation, constant low-grade infections, persistent exposure to irritants, obesity causing adipose tissue to secrete inflammatory cytokines continuously, autoimmune conditions where the immune system attacks the body's own tissues month after month, year after year. When inflammation becomes chronic, it stops being protective and becomes pathological. It contributes to atherosclerosis, narrowing blood vessels with inflammatory plaques. It promotes cancer by creating cellular environments where mutations are more likely and less likely to trigger apoptosis. It damages neurons, contributing to Alzheimer's disease. It destroys joint cartilage in arthritis. This is the immune system caught in a trap of its own making. Responses that evolve to be short and intense, stretched out indefinitely, become toxic. It's like running your car engine at maximum RPM constantly. Components designed for brief bursts of extreme stress begin to fail when that stress never stops. Evolution had no way to anticipate this. For hundreds of millions of years the pattern was simple: encounter pathogen, mount response, clear infection, or die trying, return to baseline. The idea of persistent inflammatory activation for decades at a time was simply not part of the selective pressure that shaped our immune systems. And this creates vulnerabilities that our prehistoric ancestors never faced. But inflammation gone awry pales in comparison to the cytokine storm. Cytokines, remember, are the chemical messengers that coordinate immune responses. They tell cells where to go, what to do, how aggressively to respond. In a normal, healthy immune response, cytokine signalling is like a well-conducted orchestra. Different sections coming in at precisely the right moments, volume carefully controlled, the overall effect harmonious even if intense. A cytokine storm is when that orchestra loses all coordination and every instrument begins, playing fortissimo simultaneously. It happens when positive feedback loops spiral out of control. One cytokine triggers the release of others, which trigger the release of still more, which activate more immune cells, which release more cytokines, in an escalating cascade that can go from normal to catastrophic in hours. The immune system, designed to protect, begins attacking the body's own tissues with devastating ferocity. Blood vessels become excessively permeable, blood pressure plummets, organs fail from lack of perfusion. Acute respiratory distress syndrome fills the lungs with fluid. Coagulation cascades trigger clots throughout the circulatory system, while simultaneously causing hemorrhage. This is what killed many victims of the 1918 flu pandemic. It's what makes certain Ebola infections so deadly. It's what caused many COVID-19 deaths in young, previously healthy individuals whose robust immune systems turned against them. And it represents one of evolution's most tragic trade-offs, because the capacity for a vigorous, overwhelming immune response is precisely what allowed our ancestors to survive severe infections. In a world without antibiotics, without antivirals, without modern medicine, the only way to clear certain infections was through an immune response so powerful it could overwhelm the pathogen through sheer force. Most of the time, this worked. The infection was cleared. The individual survived, perhaps scarred, perhaps weakened, but alive. Sometimes, though, the response went too far, and the immune system killed the very organism it was designed to protect. Evolution couldn't eliminate this risk without also eliminating the capacity for powerful immune responses. So the potential for cytokine storms remains, coded into our genes, a loaded gun that usually protects us but occasionally fires in the wrong direction. It's a gamble at the population level. Most individuals benefit from the capacity for strong immune responses. A small percentage suffer catastrophic immune overreaction. But because more people survive with this system than would survive without it, natural selection preserves the trait. Individual tragedy, population-level success. This is how evolution operates. Now consider one of the most elegant immunological paradoxes in biology: pregnancy. A developing fetus is, genetically speaking, a foreign organism. Half its DNA comes from the father, making it immunologically distinct from the mother. In any other context, the immune system would recognize these foreign antigens and mount an aggressive response to eliminate them. But obviously, this doesn't happen. At least, not in successful pregnancies. The placenta is an immunological marvel. a temporary organ that must perform two seemingly contradictory functions: allow nutrient exchange and waste removal, while simultaneously preventing immune rejection, all while maintaining maternal immune surveillance against actual pathogens that could harm both mother and child. How does it accomplish this? Through layers upon layers of evolutionary innovation, refined across hundreds of millions of years of mammalian reproduction. The placenta creates a physical barrier, preventing direct contact between maternal and fetal blood. It expresses proteins that suppress local immune responses, creating a zone of tolerance. It secretes factors that shift the maternal immune system away from cell-mediated immunity, the type that would attack foreign tissues, toward antibody-based immunity that can still fight infections. Regulatory T cells accumulate at the maternal-fetal interface, actively suppressing immune activation. The fetus itself produces proteins that mimic maternal immune regulators, essentially sending "don't attack me" signals continuously. But this tolerance can't be absolute, because the mother still faces pathogens that could cross the placenta and harm the developing fetus. Some of the most devastating congenital infections, toxoplasmosis, rubella, cytomegalovirus, occur precisely because the placental immune system must maintain a delicate balance between tolerance and vigilance. Tip too far toward tolerance, and infections slip through, harming or killing the fetus. Tip too far toward immune activation, and the immune system attacks the pregnancy itself, causing miscarriage or preeclampsia. This balancing act was shaped by millions of years of pathogen pressure. In our evolutionary past, women who couldn't maintain placental tolerance didn't have successful pregnancies. But women whose immune tolerance was too complete died from infections during pregnancy, or delivered children with devastating congenital infections. The system we inherited represents the survivors of this brutal selection process. It's not perfect. No system shaped by evolution ever is. But it's functional enough that our species persisted, and the mechanisms involved, the regulatory T cells, the tolerance-inducing proteins, the compartmentalization of immune responses, have become fundamental to how our immune system operates even outside of pregnancy. Learning to tolerate some things while attacking others isn't just about reproduction. It's about existing in a world full of harmless microbes, environmental antigens, and our own tissues without mounting constant destructive immune responses against everything. But before your adaptive immune system learns anything, before your innate immunity mounts any response, invaders must first breach your outermost defences. Your skin is an evolutionary fortress, not just a passive barrier, but an active defensive system refined over hundreds of millions of years since our ancestors first crawled onto land. The outermost layer, the stratum corneum, consists of dead cells packed with keratin, a tough structural protein, cemented together with lipids. It's waterproof, preventing desiccation. It's mechanically resilient, resisting abrasion and penetration. And it's constantly being shed and renewed, taking with it any bacteria or fungi that have begun to colonize the surface. But physical barriers are only the beginning. Your skin is slightly acidic, with a pH around 5.5, creating an environment where many pathogenic bacteria struggle to survive. It secretes antimicrobial peptides, small proteins that can punch holes in bacterial membranes or disrupt their metabolic processes. Sebaceous glands produce oils containing fatty acids that inhibit bacterial and fungal growth. And then there's your microbiome, trillions of bacteria, fungi, and other microorganisms that colonize your skin, competing with potential pathogens for resources and space. They're not just passive residents. They actively produce compounds that inhibit invaders, train your immune system, and maintain the skin's chemical balance. This is defense in depth, a concept refined across evolutionary time, because skin breaches are inevitable. Cuts, abrasions, insect bites, burns. Any break in the barrier creates an opportunity for invasion. So evolution layered defenses, ensuring that even when the first line fails, others remain. Mucous membranes present an even greater challenge. The linings of your respiratory tract, digestive system, and urogenital tract are moist, warm, nutrient-rich, ideal environments for microbial growth. And unlike skin, they can't form an impermeable barrier because they need to allow gas exchange, nutrient absorption, and other vital functions. So mucous membranes employ different strategies. They're covered in mucus, a complex gel containing antimicrobial proteins, antibodies, and defensive enzymes. In your respiratory tract, tiny hair-like cilia beat in coordinated waves, constantly sweeping mucus and trapped particles up and out of your lungs. In your gut, intestinal cells produce antimicrobial peptides and maintain a thick mucus layer that physically separates bacteria from the intestinal wall. Tears, saliva, and other secretions contain lysozyme, an enzyme that breaks down bacterial cell walls. They contain lactoferrin, which sequesters iron that bacteria need to grow. They contain antibodies, IgE specifically, that bind to pathogens and prevent them from attaching to cells. These barriers evolved in response to specific challenges our ancestors faced. The respiratory antimicrobial system became more sophisticated as early mammals developed higher metabolic rates and faster breathing, increasing pathogen exposure. The gut immune system evolved in response to dietary diversity and the constant bacterial exposure from consuming food. Every aspect of these barriers tells a story of evolutionary pressure, of ancestors who survived infections because their mucus contained slightly more effective antimicrobial peptides, or whose skin pH was slightly more inhospitable to particular bacteria. And these stories continue to unfold, because we're still co-evolving with our microbial environment, still adapting, still changing in response to pathogen pressure. Which brings us to perhaps the most profound immunological insight of the last few decades. We are not individuals, we are ecosystems. Your body contains roughly as many bacterial cells as human cells, possibly more. These microorganisms, collectively called your microbiome, colonize your skin, your mouth, your gut, every surface exposed to the external environment. And far from being mere passengers, they're active participants in your immune function. This relationship is ancient, truly ancient. The symbiosis between animals and their gut bacteria extends back hundreds of millions of years to the earliest multicellular life. And over that immense span of time, our immune systems and our microbiomes have become so intertwined that neither functions properly without the other. Your gut microbiome trains your immune system from birth. Bacteria colonize an infant's intestines, and immune cells learn to distinguish harmless residents from dangerous invaders. This education is fundamental. Studies of germ-free mice, animals raised in completely sterile environments, show that without microbial exposure, the immune system develops abnormally. These animals have underdeveloped lymphoid tissues, produce fewer antibodies, and respond inappropriately to threats. But the microbiome doesn't just train immunity during infancy and then step back. It continues regulating immune responses throughout life. Certain bacterial species produce short-chain fatty acids that suppress inflammation. Others stimulate regulatory T cells that prevent autoimmune responses. Some bacteria produce vitamins that support immune cell function. This is a relationship forged over evolutionary time, where our ancestors' immune systems adapted to expect and depend on microbial partners. The bacteria benefit from a stable, nutrient-rich environment. We benefit from their metabolic capabilities, their competitive exclusion of pathogens, and their immune training and regulation. But this ancient partnership creates modern vulnerabilities. Antibiotic use can devastate the microbiome, removing beneficial species and allowing opportunistic pathogens to flourish. Caesarean delivery bypasses the normal microbial colonization that occurs during vaginal birth. Modern diets may not support the bacterial diversity our immune systems evolved to expect, and when the microbiome is disrupted, immune function suffers. Rates of autoimmune diseases, allergies, and inflammatory conditions have increased dramatically in industrialized nations, a phenomenon many researchers attribute at least partially to changes in our microbial ecology. We've created environments our immune systems don't recognize. Not because we've changed, our genes are essentially identical to those of humans 10,000 years ago, but because we've changed our microbial partners, and our immune systems, expecting certain bacterial signals, receiving others, or none at all, begin to malfunction. This is evolution's time lag made visible. Our immune systems are calibrated for a microbial environment that, for many of us, no longer exists. But even beyond individual immune systems and their microbial partners, there's another layer to this story, one that operates at the population level. No two people have identical immune systems, not even identical twins. The antibody repertoires you generate through random genetic recombination are unique. Your history of infections and the immune memory cells you've developed in response is unique. Even your microbiome, shaped by your birth, diet, environment and exposures, is unique to you. This diversity is not accidental. It's a survival strategy that emerged over evolutionary time, because pathogens evolve rapidly. A bacterium can reproduce every 20 minutes. A virus can generate billions of offspring in a single infected host, and with each replication comes the possibility of mutation, the raw material for evolutionary adaptation. If every human had an identical immune system, a pathogen that evolved to evade one person's defences could evade everyone's. An infection optimised to replicate in one host would be equally effective in all hosts. Pandemic spread would be universal and catastrophic, but we're not identical. A virus that suppresses one person's interferon response might be recognised by another person's antibodies. A bacterium that hides from one person's complement system might be engulfed by another person's neutrophils. Our immunological diversity means that even novel pathogens face a heterogeneous landscape of defences. Some people in any population will be vulnerable to a new disease, but others will have immune systems that, through chance, happen to be effective against it. These individuals survive, reproduce, and pass on their genetic variants. Over generations, the population becomes more resistant. This is evolution in action, operating on human timescales. It's why some populations show resistance to diseases that devastated them centuries ago. It's why genetic variants that confer resistance to malaria or tuberculosis have spread in regions where those diseases are endemic. But here's the paradox. The immune variants that protect against one pathogen might increase vulnerability to others, or to autoimmune conditions or to allergies. Sickle cell trait protects against malaria but can cause anemia. Some HLA variants associated with strong antiviral responses are also linked to autoimmune diseases. Evolution doesn't optimize for perfection. It optimizes for survival and reproduction in specific environments. And in pathogen-rich environments, the benefits of immune diversity, both at the individual level and the population level, outweigh the costs. So we remain diverse, variably susceptible, differentially protected. Not by design, but by the relentless culling of evolutionary selection operating across hundreds of thousands of generations. This is the immune system's evolution. Not a single innovation, but a layering of defences, refined across hundreds of millions of years. From the ancient innate responses we share with invertebrates, to the adaptive immunity that emerged in early vertebrates, to the regulatory mechanisms that allow us to tolerate beneficial microbes and our own tissues, to the population-level diversity that protects our species from extinction by novel pathogens. It's a system of compromises and trade-offs, powerful enough to clear infections but capable of destroying the body in the process, tolerant enough to allow pregnancy but vigilant enough to fight pathogens, conservative enough to avoid attacking harmless microbes but reactive enough to respond to threats. And it's a system that explains so much about modern health and disease. Why autoimmune conditions have increased in sanitised environments, why some people develop cytokine storms while others barely react to the same infection, why pregnancy complications occur, why chronic inflammation contributes to diseases our ancestors rarely lived long enough to develop. Because our immune systems are still calibrated for prehistoric environments, still expecting the pathogen exposures, the microbial partnerships, the dietary patterns that shape their evolution. When we alter those conditions, sometimes for the better, sometimes inadvertently, our immune systems respond in ways that can seem paradoxical or maladaptive. But understanding this evolutionary context reveals the logic beneath the paradoxes. These aren't design flaws, they're historical adaptations, optimized for environments that no longer exist in quite the same way. And that understanding might be the key to managing the diseases that plague us today. Because you can't fix a system until you understand how it was built. And our immune systems were built, piece by piece, in response to the diseases that shaped human evolution. Which brings us to the next question. What is the legacy of those ancient diseases? How do prehistoric plagues continue to shape modern health, thousands or millions of years after the pathogens themselves have disappeared? The answer, as we're about to see, is both surprising and profound. Here's a strange irony that defines modern human existence. We live longer than any generation before us. We've conquered smallpox, tamed polio, turned deadly bacterial infections into footnotes treated with a 5-day course of pills. Our hospitals gleam with technology that would seem like magic to our ancestors. And yet, chronic diseases are everywhere. Allergies, autoimmune disorders, inflammatory conditions that turn our bodies against themselves. 1 in 5 people in developed nations suffers from allergies that would have been rare a century ago. Autoimmune diseases affect nearly 8% of the global population and are rising. Type 1 diabetes, multiple sclerosis, Crohn's disease, lupus, conditions where the immune system attacks the very body it evolved to protect. What happened? The answer lies not in what we've gained, but in what we've lost. And more precisely, in the profound mismatch between the bodies we evolved over millions of years and the world we've built in mere centuries. Welcome to the unintended consequences of survival. Let's start with a concept that's revolutionized how we think about modern disease, the mismatch hypothesis. Picture the human immune system as a standing army. Not just any army, one that's been trained, equipped, and refined through millions of years of constant warfare. Every generation that survived did so because their immune systems could handle specific threats, parasitic worms threading through intestines, bacterial invaders overwhelming the bloodstream, viral infections that could kill within days. Natural selection didn't just favour strong immune systems, it favoured immune systems calibrated to the pathogen load of the environment, active, aggressive, always ready. But here's what makes this fascinating and tragic in equal measure. That environment no longer exists. In the span of roughly 150 years, a geological eye blink, we transformed the microbial landscape of human existence. We built sewage systems, purified water, developed antibiotics, created vaccines. We didn't just defeat specific diseases, we fundamentally altered the ecosystem of pathogens and parasites that had shaped our evolution since before we were even human. And our immune systems, they're still showing up for a war that ended. Think about what this means on a cellular level. You have an immune system evolved to fight dozens of helminth species, multiple types of bacteria, and countless viral threats. It's been honed by selective pressure to be hypervigilant, quick to respond, powerful in its reactions. But in a modern, sanitized environment, those threats are largely absent. So what does a standing army do when there are no enemies to fight? It turns on its own territory. This isn't speculation. We can see the evidence in epidemiological data that's both clear and unsettling. Autoimmune diseases are dramatically more common in industrialized nations than in developing countries. Children raised on farms have significantly lower rates of allergies and asthma than urban children. Immigrant populations moving from high pathogen environments to low pathogen environments see autoimmune conditions increase within a single generation. The pattern is unmistakable. The cleaner the environment, the more our immune systems malfunction. But why? Why would an immune system that evolved to protect us become self-destructive? The answer lies in how the immune system learns what to attack. Your immune system doesn't come pre-programmed with a list of enemies. It has to learn, and that learning process requires exposure to microbes, not just dangerous pathogens, but a whole ecosystem of bacteria, viruses, fungi and parasites. Some harmful, some neutral, some actually beneficial. This exposure trains the regulatory components of your immune system, the parts that tell the attack mechanisms when to stand down, when to recognize something as harmless, when to tolerate rather than destroy. Remove those training partners, and the immune system never properly calibrates. It's like training a soldier to shoot but never teaching them to identify targets. The weapon still works, the aggression is still there, but the discrimination, the wisdom of when not to fire, that's missing. And here's where the story gets even more specific, even more personal. Remember those genetic mutations we discussed earlier? The ones that protected our ancestors from malaria, from plague, from tuberculosis. Those shields that allowed certain populations to survive when others perished. They're still here, written into the DNA of millions of people, still functioning, still shaping biology. But the diseases they evolved to fight, many of them are gone, or at least gone from the regions where these populations now live. Take sickle cell trait, the heterozygous form that protects against malaria without causing severe disease. In regions where malaria is endemic, this mutation is a survival advantage. But in North America, in Europe, in places where malaria hasn't been a threat for generations, that same mutation increases risk of kidney disease, of certain blood disorders, of complications during physical exertion at high altitude. The shield remains, but without the sword to block, it can become a liability. Or consider the CCR5 delta 13 mutation that appears to have been selected for during plague epidemics. It protects against certain viral infections. But it also may increase susceptibility to West Nile virus, and potentially influences inflammatory responses in ways we're only beginning to understand. The genetic adaptations that saved our ancestors now persist, in contexts where their benefits have evaporated, but their costs remain. Evolution doesn't have an undo button. It can only select for survival in the present moment. Once a mutation spreads through a population, it persists until it becomes disadvantageous enough to be selected against. A process that takes many generations. And in the case of humans in the modern era, where medicine can compensate for these effects, that selective pressure may never come. We're living with genetic legacies written for a different world. But let's go deeper into this mismatch, because there's a hypothesis that's both elegant and slightly disturbing in its implications. It's called the old friends hypothesis, and it reframes our relationship with parasites and pathogens in a way that challenges our fundamental assumptions about cleanliness and health. The hypothesis goes like this. Not all organisms that lived in and on our ancestors were enemies. Some were something closer to partners, or at least familiar adversaries whose presence was so constant, so evolutionarily persistent, that our immune systems actually needed them to develop properly. Helminths, parasitic worms, are the prime example. For hundreds of thousands of years, virtually every human carried intestinal parasites. Hookworms, tapeworms, roundworms. These weren't occasional infections, they were constant companions. Generation after generation, these parasites lived in human intestines, and the human immune system evolved not just to fight them, but to coexist with them. Helminths, it turns out, have a remarkable effect on the immune system. They don't just trigger inflammation, they actually produce molecules that suppress inflammatory responses. They have to. It's how they survive. A helminth that triggers too much inflammation gets expelled or kills its host, which means no more helminth reproduction. So these parasites evolve to modulate the human immune response, to turn down the volume, to create a more tolerant, less aggressive immune environment. And here's the twist that makes this so fascinating. By doing so, they were training our immune systems, teaching regulatory T cells to function, calibrating inflammatory responses, providing the very exposure that helps the immune system learn discrimination. Now remove them. Remove an entire category of organism that's been present throughout human evolution. Create an environment where children grow up without ever hosting a single helminth. What happens? Your immune system develops without that regulatory education. The volume control never gets properly installed. And when there are no real threats to respond to, that untrained, uncalibrated immune system starts reacting to things it shouldn't. Pollen, dust mites, food proteins, its own tissues. The data supporting this is striking. Multiple studies show that people with helminth infections have dramatically lower rates of allergies, asthma, and inflammatory bowel diseases. Clinical trials using controlled helminth therapy, intentionally infecting patients with specific parasitic worms, have shown remarkable results in treating Crohn's disease, ulcerative colitis, and multiple sclerosis. We spent centuries trying to rid ourselves of parasites, and we succeeded. But that success came with costs we never anticipated. The mismatch extends even further into territories that seem completely unrelated to infectious disease. Cancer. At first glance, cancer and infectious disease appear to occupy different domains. One is cells gone rogue. The other is invasion by external organisms. But the deeper you look, the more you realise they're connected through the immune system, and specifically through the evolutionary trade-offs that shaped it. Your immune system doesn't just fight pathogens, it also surveils for abnormal cells, cells that have accumulated mutations, cells that are dividing when they shouldn't, precancerous cells. The immune system can recognise these cells and destroy them before they become tumours. This is called immunosurveillance, and it's happening constantly, silently throughout your life. But here's the complication. The genes and mechanisms that make your immune system excellent at fighting infections aren't necessarily the same ones that make it excellent at detecting cancer cells. In fact, there's often a trade-off. Genes that code for aggressive inflammatory responses are fantastic when you're facing a lethal bacterial infection. They mobilize resources, they attack fast, they increase the chance you'll survive sepsis or pneumonia or cholera. But cancer cells? They're more subtle. They're you, just slightly wrong. They don't have the obvious molecular signatures of bacteria or viruses. Detecting them requires a more nuanced surveillance system, not brute force. And because natural selection operates on reproductive success, and because most cancers occur after reproductive age, there was little evolutionary pressure to optimize cancer detection. Our ancestors died from infections in their 20s and 30s. The immune system that evolved was one that maximized survival against those immediate acute threats. Cancer was someone else's problem. Natural selection didn't care about it. So we inherited immune systems optimized for a problem set that no longer dominates. We live long enough now for cancer to matter, but our immune systems are still playing by prehistoric rules. This helps explain why certain immune gene variants associated with strong responses to infections also correlate with increased cancer risk. The same inflammatory pathways that fight pathogens can create environments that promote tumour growth. The same genes that saved your ancestors from dying of tuberculosis at 25 might increase your risk of colon cancer at 65. Evolution doesn't optimize for longevity, it optimizes for reproduction, and those are very different things. But here's where this story shifts from academic curiosity to immediate practical relevance. Understanding the genetic variations that prehistoric diseases left in different populations isn't just historical interest, it's predictive power for future pandemics. When COVID-19 emerged, one of the most puzzling aspects was the dramatic variation in severity across populations. Some people experienced mild symptoms, others died. Age was a factor, underlying conditions mattered. But there was also genetic variation that tracked in some cases with ancient evolutionary pressures. Researchers discovered that some genetic variants associated with severe COVID-19 outcomes showed signals of positive selection in the past, meaning they were once advantageous against some other pathogen. The specific advantage has been lost to time, but the gene variant remains, now creating different vulnerabilities. Other populations carry genetic profiles that seem to offer some protection, possibly because their ancestors faced different disease pressures that selected for different immune responses. This isn't about race, it's about population history, and the specific pathogen landscapes different groups encountered over thousands of years. It's about understanding that immunity isn't universal, it's shaped by ancestry and the diseases that shape that ancestry. And this understanding has profound implications. When we develop vaccines, when we predict who will need intensive medical intervention, when we allocate resources during a pandemic, population genetic variation matters. A vaccine that produces robust responses in one population might produce different responses in another because of immune system differences rooted in prehistoric disease exposure. We're learning to read the book of human genetic variation, not as a catalogue of differences, but as a medical history. A history written by disease, recorded in DNA, and directly relevant to health challenges today. And speaking of pandemics and diseases that refuse to disappear, some of our prehistoric adversaries are still here. Tuberculosis kills 1.5 million people every year. Yersinia pestis, the plague bacterium, still causes outbreaks. These aren't diseases we fail to eliminate. They're diseases we can't eliminate, at least not easily, because of how deeply they've co-evolved with humanity. Tuberculosis has been with us for at least 70,000 years, probably longer. It appears in ancient Egyptian mummies, in Peruvian mummies, and skeletal remains across continents. It has evolved alongside us, adapting to our immune responses, finding ways to persist inside our own immune cells, going dormant when conditions are unfavourable, and reactivating when we're vulnerable. A third of the global population carries latent tuberculosis infection, not active disease, just the bacteria, dormant, waiting. Most will never develop active TB, but some will, when age or HIV or malnutrition weakens their immune systems. You can't eradicate an organism that hides inside a third of humanity. And plague? Even after the Black Death killed half of Europe, Yersinia pestis persisted in rodent populations across Asia, Africa, and the Americas. It causes thousands of cases annually, even today. We have antibiotics. We have public health infrastructure. but we can't eliminate a pathogen that has ecological reservoirs completely independent of human populations. These diseases have outlasted empires. They've seen civilizations rise and fall. They've adapted to every weapon we've developed against them, and in a sense they've earned their persistence. They've been selected over and over for the specific traits that allow them to survive in human bodies, in human populations, in the ecological systems we inhabit. They're not just parasites. They're specialists in the biological niche called humanity. Which brings us to one of the most exciting frontiers in this entire story, ancient DNA. We're not just studying modern diseases anymore. We're extracting pathogen DNA from archaeological remains, from medieval plague victims, from Bronze Age skeletons. We're sequencing the genomes of bacteria and viruses that killed people thousands of years ago. And what we're learning is rewriting our understanding of infectious disease history. We've discovered that the plague bacterium went through multiple genetic changes over centuries, becoming more virulent, then less virulent, adapting and evolving in response to human populations. We found that tuberculosis strains from pre-Columbian America were different from European strains, suggesting independent evolution and cross-oceanic transmission long before European contact. We've identified smallpox DNA in Viking-era remains, pushing back the timeline of that disease in Europe. We've traced the origins of syphilis, of leprosy, of typhoid through genetic analysis of ancient pathogens. But more than just historical curiosity, this research teaches us how pathogens evolve, how they adapt, what genetic changes make them more dangerous, what ecological or population factors drive their evolution. And all of this is immediately relevant to emerging infectious diseases today. When a new pathogen appears, whether it's Ebola or Zika or the next coronavirus, we need to predict how it will evolve. Will it become more transmissible, more lethal? Will it adapt to human immune responses? Ancient pathogen DNA provides a record of how these processes have played out in the past. It's an evolutionary playbook. And by studying it, we're learning to anticipate what might come next. We're essentially reading the deep history of human pathogen evolution and using it to prepare for the future. Each ancient genome is a lesson in microbial adaptation. Each plague victim's DNA is data. It's archaeology as epidemiology, history as predictive medicine. And now we arrive at perhaps the most philosophically challenging question in this entire narrative. What happens next? For millions of years, disease was a selective force. People with genetic variants that protected against infection survived. Those without them died. The population evolved. Generation by generation, humans became more resistant to the diseases that surrounded them. But modern medicine changes that equation fundamentally. A child born with an immune deficiency that would have been fatal a century ago can now survive with treatment. A genetic variant that would have led to death from a childhood infection is now medically manageable. People who would never have survived to reproduce in the prehistoric world now have children, passing on genetic variations that would previously have been eliminated by natural selection. This isn't a value judgment, it's one of humanity's greatest achievements. We've removed disease as an agent of natural selection. We've chosen compassion over evolutionary pressure, but it does raise questions. If we're no longer being shaped by infectious disease in the same way, how does human evolution proceed? Are we halting our own evolutionary adaptation to pathogens? And if so, what does that mean when new diseases emerge? The answer is complex. We haven't stopped evolution. We've changed what drives it. We've shifted selective pressures from natural to cultural, from environmental to medical. But simultaneously, we've created new pressures on pathogens. Every antibiotic we deploy selects for resistant bacteria. Every vaccine we develop forces viruses to evolve new variants. Every medical intervention creates an evolutionary pressure on the organisms we're trying to control. We're not ending the evolutionary arms race. We're accelerating it and changing the battlefield. The pathogens are still evolving. Antibiotic resistance is spreading faster than we can develop new drugs. Viruses are adapting to vaccines. Fungi are becoming resistant to antifungals. And here's the uncomfortable reality. Pathogens evolve faster than we do. They have shorter generation times, larger population sizes, higher mutation rates. They can adapt in years to pressures that would take us millennia to respond to genetically. So the evolutionary future of human-pathogen relationships isn't about us evolving to resist them. Not anymore. It's about our technology evolving faster than they do. It's an arms race between microbial evolution and human innovation, and the stakes have never been higher. So here we are, descendants of survivors. Every person alive today carries genetic echoes of diseases that killed billions, the mutations that protected against malaria, the immune variants selected by plague, the inflammatory responses calibrated by millennia of parasitic infections. We are not blank slates, we are accumulations, layers upon layers of evolutionary responses to threats our ancestors faced. And those layers don't always serve us well in the world we've built. The shield can become a burden, the sword can cut its wielder, the immune system can turn on itself. But understanding this, really understanding the deep history written in our genes and the evolutionary context that shaped them, gives us something powerful. It gives us perspective on modern disease. It helps us understand why certain populations are vulnerable to certain conditions. It guides vaccine development and pandemic response. It suggests new treatments for autoimmune disorders and allergies. It reminds us that we are not separate from nature, not exempt from evolutionary rules, no matter how advanced our technology becomes. We are still animals, still shaped by selection, still carrying the biological legacy of every challenge our lineage faced. And perhaps most importantly, it reminds us that the dance between humanity and disease isn't over. It's never over. It's an ancient rhythm that predates our species and will continue long after us. We're just the current dancers, trying to keep pace, trying to survive the next song. And so far, against odds that would have seemed impossible to any of our ancestors, we're still dancing. Conclusion: the endless dance between humanity and disease. And so, we arrive here. At the end of a journey that began millions of years ago, when our ancestors first encountered invisible enemies they couldn't name, couldn't see, couldn't understand, but somehow, against astronomical odds, survived. We've traced the arc from those first agricultural diseases, born in the mud and grain of our earliest settlements, through the genetic scars that still live in our DNA, HLA diversity, sickle cell, lactose tolerance, to the plagues that carved populations like sculptors shaping stone. Each disease left its mark. Each outbreak rewrote the genetic text of humanity. And here's the humbling truth we've been circling this entire time. We don't exist despite diseases. We exist because of them. Every infection was a test, every plague a filter. The pathogens shaped us as much as tools or fire or language. They made us who we are. This story hasn't ended, by the way. COVID-19 wasn't an aberration. It was the latest verse in humanity's oldest song. Future pandemics will come. New diseases will test us, and our immune systems, our genetics, our behaviours, they'll continue to adapt. The dance never stops. It only changes tempo. We're fortunate to live in this brief, miraculous window where modern medicine can intervene. Antibiotics, vaccines, treatments that would seem like magic to our ancestors. But zoom out far enough and you'll see it's just a moment, a breath in the long evolutionary conversation between humanity and disease. What does this perspective teach us? Perhaps that resilience isn't about never falling. It's about rising, adapting, changing. That survival isn't a solo act. It's written in community, in diversity, in the collective strength of shared immunity and shared struggle. If you've enjoyed this journey through deep time, I'd be grateful if you'd like and subscribe. There's more to explore about human evolution, about the forces that shaped us. And to all of you watching from around the world, thank you for being here, for sharing where you're from in the comments. We're all connected by this ancient story. Rest well tonight, knowing you're part of something vast, something that began long before you and continues far beyond. Sweet dreams.